Good morning, everyone. We will just give it 2 more minutes just to allow everyone to join before we get started. Thank you. Okay, I think we can get started in the meantime. So good morning, everyone, and welcome to today's webinar titled Endotoxin Testing Simplified, Reliable Detection and Best Practices for the Laboratory. So thank you for taking the time to join us, whether you're in the pharmaceutical or biotechnology research industry or quality control. I'm sure you will find something very useful in this webinar. Today's presenter is Dr. Daniela Da Silva, who is an application specialist at Whitehead Scientific. Daniela works within the molecular and cell biology portfolio, and her work focuses on translating complex scientific concepts into practical lab solutions. So this makes her ideally placed to guide us through today's topic. During this webinar, Daniela will cover the principles of endotoxin detection, common sources of variability, strategies to improve reliability and consistency, and practical considerations for optimizing your workflow. We will allow questions at the end of the presentation, so please feel free to submit any questions you have during the webinar. The chat function is open throughout the session, or you can ask your questions at the end. So without further ado, I'd like to hand over to Dr. Daniela Da Silva.
Thank you, Sanisha. Good morning, everyone. I'm just going to get into sharing. There we go. Can everyone see my screen? Perfect. Okay, good morning, everyone. Thank you so much for joining today's webinar. As Sanisha mentioned, today's session is called Endotoxin Testing Simplified, the reliable detection and best practices for the laboratory. So just before we begin, I'd like to briefly walk through the structure of today's session. We'll start by discussing what endotoxins are and why they continue to be important contaminants within pharmaceutical and biotechnology environments. We'll then move into the major endotoxin detection technologies and look at the biological principles behind each of the assays. Once we've covered the detection methods, we'll discuss regulatory considerations, including the endotoxin limits, some of the key calculations that laboratories perform routinely. And then from there, we'll look at common sources of variability, why they seemingly identical samples can sometimes produce different results. And finally, we'll discuss best practices for improving consistency and explore the practical challenges for simplifying endotoxin testing workflows. So before discussing testing methodologies, I always find it useful to establish exactly what we're trying to detect. Endotoxins are lipopolysaccharides. They're often referred to as LPS molecules that form part of the outer membrane of a gram-negative bacteria. So structurally speaking, endotoxins consist of three main regions. You have the O antigen, which extends outward from the bacterial surface. You have a core polysaccharide region, and then you have lipid A, which is the biologically active portion of the molecule. So lipid A is particularly important because it is responsible for the pyrogen and the inflammatory effects that are associated with endotoxin exposure. So one important point to remember is that endotoxin contamination and bacterial contamination are not necessarily the same thing. I find sometimes people do tend to confuse that. So a product may be completely sterile and free from viable microorganisms, but they can still contain endotoxins that will remain even after bacterial cells have been either removed or even destroyed. So this is one of the reasons why endotoxin testing remains such a critical part of pharmaceutical quality control. One of the challenges with endotoxin testing is that endotoxins do not behave like simple molecules that are dissolved uniformly in a solution. So these are amphiphilic molecules, meaning that they contain both hydrophobic and hydrophilic regions. Because of this, endotoxins naturally interact with each other and with their environment. So they can form aggregates and micelles, which then also bind to proteins. They absorb onto surfaces and they interact with components of the sample matrix. Their behavior can also change depending on factors such as pH, ionic strength, temperature, surfactants, and sample composition. So what does this mean in practice? What it means is that two samples containing exactly the same endotoxin concentration may not necessarily behave the same way during testing. In many cases, what we measure is not simply endotoxin concentration, but technically endotoxin accessibility. And this is where much of the analytical complexity associated with endotoxin testing really begins. Endotoxins are among the most potent pyrogens encountered in pharmaceutical manufacturing. Even relatively low levels can trigger significant biological responses when they're introduced to the body. So once endotoxins enter systemic circulation, they activate components of the innate immune system, and this then stimulates inflammatory signaling pathways. Clinically, what this means is that it can result in fevers, inflammation, hypertension, and in severe cases, could even introduce shock or organ dysfunction. From a laboratory perspective or even a manufacturing perspective, endotoxins can also have a substantial impact. They will affect cell culture performance, they influence experimental outcomes, they reduce reproducibility, they create challenges during product release. So ultimately, monitoring endotoxins is about more than just a regulatory compliance. It's about protecting the patient's safety, it maintains the product's quality, and it ensures confidence in the manufacturing and research process. So when laboratories encounter endotoxin contamination, it's natural to focus immediately on the assay itself. However, contamination often originates much earlier in workflow. So water systems tend to be the most common sources of endotoxin contamination, and they remain critical as a critical area of control in pharmaceutical manufacturing. Raw materials, excipients, buffers, and process components also can introduce endotoxins within a workflow. In addition, consumables such as plasticware, your filters, your tubes, even your pipette tips can contribute if they are not appropriately controlled. Environmental factors, operator handling, manufacturing equipment, and even routine laboratory practices all play a significant role in decreasing endotoxin contamination. A useful principle to remember is that preventing endotoxin introduction is usually much easier than having to now remove the contamination later. For that reason, effective contamination control starts with a good workflow design and a proactive risk management. There are multiple technologies available for endotoxin and pyrogen testing, and each of these measures a slightly different biological response. Historically, we had the rapid pyrogen test, which evaluated whole animal responses to pyrogen exposure. However, this test has now been systemically phased out. More recently, we have the monocyte activation tests, or the MAT assays, which provide an in vitro approach that measures cytokine production. You have your luminous amoebocyte lysate or your LUL assay, and this is to detect the activation of the horseshoe crab coagulation cascade. And more recently, we have the recombinant factor C technologies, which focus specifically on the endotoxin recognition step within that particular cascade. So, although these methods are often discussed together, I feel it's important to recognize that they are not measuring exactly the same biological phenomenon. So understanding that there are those differences helps laboratories then select the most appropriate technology for their specific application. Before discussing the different LUL methodologies, it's always helpful to understand where the technology originates from. So LUL stands for the luminous amoebocyte lysate, as I mentioned. It is derived from the blood cells known as amoebocytes of a horseshoe crab. These animals possess a highly sensitive innate defense mechanism. So when an endotoxin is detected, a coagulation cascade is rapidly activated to help contain potential microbial threats. Scientists then recognize that this natural defense mechanism could then be adapted into an analytical tool for detecting bacterial endotoxins. What makes this system particularly remarkable is its sensitivity. Even extremely low concentrations of endotoxin can trigger the cascade, making it one of the most sensitive biological detection systems that we have available. This biological amplification is what forms the foundation of the traditional endotoxin testing. It is important to note that while traditional LUL assays have served the industry exceptionally well for decades, biological systems naturally introduce certain challenges. So, because lysate is derived from a biological source, there does sometimes happen to be variability, which occurs between reagent lots. Supply considerations and sustainability concerns have also become increasingly important topics within the industry itself. In addition, traditional coagulation cascade contains multiple biological components, and this is where pathways such as whilst going into the pathways, it may respond to substances such as beta glucans, which is naturally found within these type of biological systems. This then sometimes contributes to unwanted signal generation in certain sample types. So it is important that these limitations do not diminish the value of the LUL testing. It is important to acknowledge that there are some limitations to this. Now that we've discussed where LUL technology originates, I feel let's look a little bit more closely into the biological mechanism that makes these assays so sensitive. So at the center of the process is the endotoxin recognition. So when an endotoxin is present, it will activate a protein known as factor C. This is the initial recognition event and it is essential as it serves as the trigger for the entire cascade going forward. So once your factor C is activated, it activates factor B, which then turns and activates downstream clotting enzymes. These enzymes is what generates a measurable response, whether that's a clot formation, increase in turbidity, color development, or even a fluorescence signal, depending on the different assay format that will be used. What makes the system so powerful is the concept of biological amplification, as mentioned earlier. So a very small amount of endotoxin can initiate a series of reactions that produces a much larger detectable signal. So this amplification is what allows the LUL-based assays to achieve such impressive sensitivity. However, it is also important to remember that because multiple steps are involved, interference occurring anywhere within the cascade can potentially affect the assay performance, as also mentioned earlier. So let's start with the simplest and most established LUL methodology. This is the gel clot assay. So the principle is relatively straightforward. You have a sample that is mixed with the LUL reagent, and it is then incubated under controlled conditions. If endotoxin is present within the sample, or above a specific sensitivity, the activation of the coagulation cascade results in the formation of a stable gel clot. So after incubation, the reaction vessel is gently inverted. If the clot remains intact, the sample is then considered positive. If there is no stable clot that forms, then the result is considered negative. So one of the major advantages of using the gel clot testing assay is its simplicity. It uses very little instrumentation and it's an accessible method for numerous laboratories. It is also a very long established method with a strong regulatory history. However, because the endpoint relies on visual interpretation, some degree of subjectivity does occur, and this is particularly with your borderline samples. So, in such, it's also a qualitative method and it's not fully quantitative. And for this reason, not many laboratories tend to stick or stay with this assay. And instead, they transition toward a kinetic method when quantitative data or even a higher throughput within the lab is essentially required. And that leads us to the kinetic turbidimetric assay. Importantly, all these assays use the same biological cascade that we've already discussed, so the difference really lies in how the response is measured. So as the coagulation cascade progresses, insoluble coagulant complexes begin to form within the reaction mixture. As these complexes accumulate, the solution will then become increasingly turbid or even cloudy, as some would describe it. Rather than waiting for final endpoint, the instrument will continuously monitor changes in turbidity over time. Typically, this measures absorbance at 340 nanometers. The key question being asked with this particular assay is how quickly is turbidity developing? So higher endotoxin concentrations within a sample will result in faster activation of the cascade and therefore a more rapid increase in turbidity. The assay software will then compare the reaction kinetics to a standard curve, and it calculates the endotoxin concentration like that. One of the strengths of the kinetic turbidimetric assays is their broad dynamic range and the fact that it is a quantitative output. So these characteristics make them particularly attractive for routine quality control environments. However, it is important to note that the assay, because it relies on optical measurements, samples that are already turbid or contain some sort of particulate material within the sample may require a different assay, as this would cause interference. So of the various L methodologies, the kinetic chromogenic assay tends to be the most widely used in modern pharmaceutical endotoxin testing. Like the previous methods, the assay still begins with the endotoxin activation of the Lal cascade. However, instead of measuring now a clot formation or turbidity, we're measuring the fact that the activated clotting enzymes will cleave to synthetic chromogen substrates. So this chromogen substrate, normally it's called a p-nitrogen or PNA. The enzyme will then cleave to it and then it releases this yellow chromophore. The instrument will continuously monitor the color development over time. Typically, this occurs at 405 nanometers. As with the turbidimetric assay, the speed of the signal generation is directly related to the endotoxin concentration that is present within your sample. So a higher endotoxin concentration will produce a faster color or yellow color development and shorter reaction times. The reason that the chromogenic assays tend to be so popular is that they combine the excellent sensitivity with a quantitative output and a compatibility with a large variety of samples. So these characteristics make them particularly well suited to pharmaceutical quality control laboratories, where factors such as consistency, their throughput, and their reproducibility are very important priorities. Then we have recombinant factor C, or RFC, which has become an increasingly important option within modern endotoxin testing. Unlike the traditional LUL assays, which rely on a complex lysate extracted from the horseshoe crab amoebocytes, RFC assays focus specifically on the endotoxin recognition step of the cascade. So the assay uses recombinant version of the factor C produced through biotechnology processes. So when endotoxin is present, it binds to and activates the recombinant factor C. The activation then leads to the cleavage of a fluorogenic substrate here, and that then generates a measurable fluorescent signal. The amount of fluorescence produced is directly related to the endotoxin concentration that is present within the sample. Results will then be quantified against a standard curve, much like the other kinetic endotoxin assays. One of the major advantages of this approach is that it reduces biological complexity by focusing specifically on the endotoxin recognition mechanism itself. So because the assay does not contain the additional components present in traditional lysate-based systems, it eliminates that beta-glucan reactivity and it provides high specific endotoxin detection assay. In addition, recombinant technology supports the long-term supply stability and sustainability initiatives that are occurring within this field, and it maintains the sensitivity required for the routine endotoxin testing. So as a result, many laboratories are now evaluating RFC as part of both current and perhaps even future endotoxin testing strategies. Whenever new analytical technology is introduced, one of the first questions laboratories tend to ask is, has this approach been successfully implemented in a real-world regulated environment? So over the past several years, RFC has moved well beyond being considered an emerging technology. A growing number of pharmaceutical and biological organizations have successfully implemented RFC-based endotoxin testing within their already regulated workflows. The publication that I've highlighted here on the slide is an example of how RFC has been evaluated and applied within pharmaceutical development and quality environments. The findings demonstrate that RFC can provide equivalent endotoxin detection performance while still supporting the broader sustainability and supply continuity objectives that are occurring within the field. Importantly, the adoption of RFC or recombinant technologies is driven not only by sustainability considerations, but also by scientific and operational benefits. These include the fact that the assay is highly specific, workflow consistency, and reduced biological variability. As regulatory agencies and pharmacopoeial bodies continue to evaluate and support alternative approaches, many organizations are tending to incorporate the recombinant technologies into their long-term endotoxin testing strategies. So what we're seeing across the industry is a gradual shift from asking whether RFC is viable to asking where and how it can actually be best implemented within their existing workflows. So now that we've looked at the major assay formats individually, a question I often receive is, which endotoxin assay should we choose? And the reality is that there's really a single assay that is universally best for every application. Instead, assay selection should be driven by the specific need of that particular laboratory as well as the workflow requirements within that laboratory. So as you can see from the table, each methodology offers its own strengths. Your gel clot assays are simple, they're very well established, and they require minimal instrumentation, but they do provide qualitative data. They are also less suited to high throughput laboratories or environments. You have your kinetic turbidimetric assays, which offer a quantitative measurement and are a broad dynamic range, and they're useful for many QC applications. However, they are limited to the samples that you'd be working with. Kinetic chromogenic assays will provide excellent sensitivity, their quantitative results, and they're particularly well suited to automation and high throughput testing environments, as well as a broader variety of sample types. Then you have your recombinant factor C assays, which also provide highly sensitive quantitative measurements while still focusing specifically on the endotoxin recognition through that factor C activation. So when selecting a method, it's important to consider factors such as what is the required sensitivity? What is our sample throughput? What are our automation requirements? Do we have the available instrumentation? What are our regulatory expectations? And what are our long-term workflow objectives? Are we planning on moving into maybe incorporating RFC or a higher throughput within our lab? So rather than just asking which method is superior, I would rather encourage you to ask which method best fits our product, our workflow, and our testing strategy currently. Ultimately, the best assay is the one that consistently delivers reliable, reproducible, and scientifically defensible results within your specific testing environment. Now that we've discussed the major assay technologies, it is important that I address the regulatory framework that supports endotoxin testing. So within the European Pharmacopoeia, Chapter 2.6.14 describes bacterial endotoxins test and outlines the accepted methodologies for endotoxin testing. However, pharmacopoeial expectations extend beyond simply just performing an assay. So a recurring theme throughout the regulatory guidance is suitability. In other words, laboratories must demonstrate that the method that they're performing is appropriate for the specific product that is being tested. So this includes evaluating potential interference, confirming acceptable recovery, understanding the assay limitations, and validating the overall testing strategy. Regulators are interested not only in whether testing was performed, but whether the testing approach is scientifically appropriate and capable of generating reliable results. Ultimately, compliance is about demonstrating the confidence in the testing process as well as the data that is being generated. So having discussed detection technologies, the next logical question is, how much endotoxin is actually acceptable. So the answer begins with understanding threshold pyrogen doses. These values represent the maximum amount of endotoxin exposure that is considered acceptable for a patient under specific circumstances. So for standard parenteral products, the accepted threshold is typically 5 endotoxin units per kilogram of body weight per hour. For intrathecal products, where administration occurs directly into the central nervous system, the limit becomes significantly lower at 0.2 endotoxin units per kilogram. Medical devices do have their own defined limits based on a route of administration as well as the intended use. So these limits are important because what they ultimately do is determine the sensitivity requirements of the analytical methods that we use. So in other words, endotoxin limits provide a framework that defines how sensitive our testing strategy should be. So once threshold pyrogen dose values have been established, we calculate the endotoxin release limit for a specific product. The equation is actually quite simple. So you have your endotoxin release limit, or your ERL, and that is calculated by dividing K by M. What K is it represents your threshold pyrogen dose defined by the pharmacopoeia. You have M, which represents your maximum dose of the product that a patient may receive over a specified period of time. So let's consider a simple example over here. If K equals 5 endotoxin units per kilogram per hour, and the maximum dose is, say, 10 milliliters per kilogram per hour, your endotoxin release limit would then be 0.5 endotoxin units per milliliter. And this value becomes critical as a parameter during method development, as it defines the level of endotoxin that must be reliably detected as well as controlled. So understanding the release limit is therefore fundamental to then selecting the appropriate testing strategy and demonstrating its compliance. One of the most important calculations encountered during endotoxin method development is your maximum valid dilution, or your MVD. So MVD defines the greatest dilution that can be applied to a sample while still allowing the assay to detect endotoxin at a required release limit. So this concept becomes particularly important when dealing with challenging sample matrices. Dilution is often one of the simplest and the most effective approach for reducing matrix interference. However, dilution cannot be increased indefinitely. Eventually, an endotoxin concentration becomes too low for the assay to detect reliably and therefore cannot be used. So the MVD calculation establishes the boundary between acceptable dilution and loss of required sensitivity. In this example over here, an endotoxin limit of 0.5 endotoxin units per milliliter and an assay sensitivity of 0.005 endotoxin units per milliliter will result in an MVD of 100-fold. Understanding this value helps laboratories then balance the interference reduction with analytical sensitivity. One of the most common questions laboratories tend to ask is, why do my endotoxin results sometimes vary when I don't think anything has changed? So the reality is that assay chemistry is often only one part of the story. Variability can originate from multiple sources throughout your workflow. Sample matrices may contain substances that interfere with the assay. Then you have environmental factors such as airborne particles, water quality, or laboratory conditions, which can also then contribute to these inconsistencies. You have operator technique, pipetting accuracy, timing, and sample handling. All of this can influence results. Instrument performance, calibration status, and temperature control are equally important. Even something as simple as your reagent storage conditions or the quality of consumables can have a measurable impact. When we think about endotoxin testing, it's always helpful to view it as an entire process rather than just the single assay. The more potential sources of variability we can identify and control, the more confidence we can have in the results that we're now generating. One of the most important concepts in practical endotoxin testing is interference. So interference generally falls into two categories: your inhibition and your enhancement. So what inhibition is it occurs when components within the sample suppress the assay response. As a result, endotoxin may be present, but the assay detects less than what is actually in the sample. So this can potentially lead to false negative results, which are particularly concerning from a product safety perspective. And then you get enhancement, which is actually the opposite. So in this case, sample components will amplify the assay response and create an appearance of an even higher endotoxin level than what is actually present within the sample. So both scenarios can lead to misleading conclusions. This is why suitability testing is a very critical component of endotoxin method validation. Ultimately, before we can trust an assay result, we need to demonstrate that the sample matrix itself is not influencing the assay's ability to detect the endotoxin accurately. This then brings us directly to product suitability testing. So at its core, product suitability asks a very simple but extremely important question. Can this assay reliably detect endotoxin in this specific product matrix? So to answer that question, known concentrations of endotoxin are introduced into the sample and recovery is evaluated. If recovery falls within the accepted range, we gain confidence that the assay is functioning appropriately in that matrix. If the recovery is too low, inhibition may be present, and if the recovery is too high, we may have enhancement occurring. So one important point to remember is that performance demonstrated in water does not automatically translate to performance in a in a complex product formulation. So every matrix will behave differently. This is why product suitability testing remains one of the most important exercises in establishing confidence in your endotoxin testing method. Once we understand the sources of variability, the next step is then implementing practices that improve the consistency. So reliable endotoxin testing is rarely achieved through assay chemistry alone. Instead, reliability comes from controlling the workflows surrounding your assay. This includes standardizing your sample preparation procedures, maintaining consistent incubation conditions, using appropriate consumables, and ensuring analysts are properly trained. Attention to detail is particularly important. So small inconsistencies that seem insignificant at the time or on their own can accumulate and then ultimately affect the assay performance. One principle I often share with laboratories is that reproducibility is usually a reflection of process control. The more standardized and controlled the workflow becomes, the easier it is to generate reliable and reproducible results over time. So as testing volumes increase, laboratories naturally begin looking for ways to now improve their efficiency. However, increased throughput often introduces additional complexity. Manual calculations, data transcription, spreadsheet manipulation, and result interpretation all create opportunities for variability and human error. And this is why many laboratories then move toward more integrated and standardized workflows. The goal is not simply to save time. Your goal here is to reduce unnecessary variables that can influence data quality and the reproducibility. So when workflows become simpler and more consistent, laboratories can then often find that both their efficiency as well as their confidence in their results improve simultaneously. In many cases, simplification is actually one of the most effective strategies for improving overall assay robustness. And that leads us to this. As endotoxin testing workflows become increasingly quantitative, instrumentation now plays a much more significant role in ensuring the reliability and the reproducibility of our results. So the ideal platform should not only support the assay formats being used, but also provide consistent environmental control. It has to give accurate data acquisition and streamlined data analysis. One of the conversations I have quite frequently with customers is around selecting the right detection platform, not only for their current testing requirements, but also for where they see their endotoxin testing moving in the future. Different assay formats have different detection requirements. So traditional kinetic chromogenic and kinetic turbidimetric assays rely significantly on absorbance measurements, while recombinant factor C will require fluorescence detection. So this is where platform flexibility becomes important. The Lonza Nebula Absorbance Reader supports traditional LUL methodologies, including your kinetic chromogenic and your kinetic turbidimetric assays, making it well suited to laboratories focused on your conventional endotoxin testing workflows. And then for laboratories looking to incorporate both your traditional LUL assays and your recombinant factor C methodologies, would then require your Lonza Nebula multimode reader. This provides both the absorbance and fluorescence detection capabilities within one platform. So in addition, it's also important to note that both these systems do support monocyte activation testing, providing the flexibility for laboratories working across multiple pyrogen testing strategies. Ultimately, Instrumentation should do more than just simply generate the data. It needs to help your laboratory improve its consistency, support the reproducibility, and simplify workflow management. This then provides the confidence in the results that are being generated. As laboratories become increasingly digital, data management becomes just as important as assay performance itself. So generating accurate results is only one part of the process. Laboratories must also be able to demonstrate traceability, consistency, and the data integrity. Modern software platforms help support these requirements through automated calculations, they standardize templates, they provide audit trails, and electronic record of management. By reducing manual data handling, laboratories can then minimize transcription errors and improve the overall workflow consistency within their lab. So, trend analysis capabilities can also provide valuable insight by identifying gradual shifts in performance before they become significant problems. Ultimately, effective software should simplify your data management and it should support the compliance rather than creating additional administrative burden. So before we move to the conclusion, I'd like to just reinforce one key message from today's discussion. It's about reliable endotoxin testing is that it's not simply about selecting the assay. It's about understanding and controlling the entire workflow. So sample preparation influences your assay performance. Your assay selection then influences the sensitivity and the suitability. Your instrumentation will influence your reproducibility. Your software will influence your traceability and your data integrity. So when laboratories experience challenge, the root cause often lies at the interface between these workflow components rather than just at a single step. This is why integrated approaches become increasingly valuable. By viewing endotoxin testing as a complete workflow rather than just a standalone assay, laboratories can often achieve greater consistency, efficiency, and confidence within their results. So as we reach the end of today's session, I'd like to briefly summarize the key messages we've discussed. First, endotoxins remain analytically challenging molecules, not because the assays are inherently difficult, but because endotoxins themselves behave differently depending on their environment and the sample matrix in which they are present. Second, there is no single endotoxin assay that is best for every application. Method selection should always be guided by factors such as the sample type, the required sensitivity, the throughput needs within the lab, workflow requirements, and regulatory expectations. Third, product suitability testing remains essential. Before we can trust an endotoxin result, we need to demonstrate that the assay performs reliably within the specific sample matrix that it's being tested in. Fourth, many of the sources of variability encountered in endotoxin testing are manageable when laboratories implement robust, standardized, and well-controlled workflows. And finally, simplifying workflows often improves both the efficiency and the reliability of reducing opportunities for variability and even human error. If there's one message I'd like you to take away from today's webinar, it's this. Reliable endotoxin results depend on reliable workflows. The ultimate goal is not simply to generate data, but to generate results that are accurate, reproducible, and scientifically defensible, results that laboratories can trust with confidence. That brings us to the end of today's webinar. I hope today's discussion has provided a practical perspective on endotoxin testing and highlighted some of the factors that contribute to reliable and reproducible results. Thank you all for your time. I'd be happy to answer any questions if there are.
Okay, thank you, Daniela. I think we do have Bitsy and hand up. Fatima, do you have a question? No. Okay. No problem. Okay. You can type it in the chat box if you wish. Let me just open up the chat box quickly. Any questions? Anyone wants to type it in? That's fine as well. Okay, we have a question regarding software, Daniela. Fatima, is it regarding the actual program or how to use the software? Okay. Daniela, the question is, can you make changes to your parameters to make it product specific?
I believe that you can. I'll have to look into how exactly you can go about doing that. Is that from Fatima?
Yes.
So Fatima, what I can do for you is I will then draft you a mail. Let me look a little bit into it, but I'm pretty sure that you can.
Okay. We also have another question from Sasha regarding the price of the multimode reader. Sasha, we do have your details. I am able to provide further information to you via email. So I can get back to you today still. Okay. Dion Adams, what is the price difference between the Lonza base model and RFC analysis model? Can the base model be upgraded to the RFC? So if you're using the multi-mode reader, yes, you can alternate between absorbance and fluorescence. So the RFC uses fluorescence. If you are using the absorbance reader, you cannot do RFC detection on that. I hope that answers your question. The price difference, I can respond to you via email. I just have to double check pricing between the absorbance and the multi-mode reader. So I'm happy to follow that up via email. Okay. Are there any other questions? Okay, then I think that is it. Thank you everyone for joining. The webinar will be available on the Whiteside web shop if you do want to share with your colleagues at a later stage. Have a lovely rest of your day and thanks once again. Bye.